हॅलो फ्रेंड्स, राणा अकॅडमी या यूट्यूब चॅनलवरती आपणा सर्वांचं हार्दिक स्वागत आहे. आपण स्टार्ट केली आहे बायोलॉजी 11th क्लास महाराष्ट्र स्टेट बोर्डची. त्यामध्ये आपण आतापर्यंत 5th चॅप्टरपर्यंत पाहिलं. त्यानंतर आपण आता पाहणार आहोत 6th नंबरचा चॅप्टर आपला बायोमॉलिक्युल्स. चला तर मग तुम्ही या चॅनलवरती नवीन आला असाल तर प्लीज या चॅनलला सबस्क्राईब करून ठेवा आणि बेल आयकन हिट करा, जेणेकरून तुम्हाला आमचे नवीन व्हिडिओ पाहता येतील. चला तर मग व्हिडिओला सुरुवात करूया. 6 नंबरचा आपला चॅप्टर आहे Biomolecules. Our planet is having a wide diversity of living organisms that are classified as unicellular, consisting of single cell, including many cells, example plants and animals. You have also learned that living organisms have cell as the basic structural and functional unit. The cells have protoplasm containing numerous chemical molecules, the biomolecules. Biochemistry is biological chemistry that provides us the idea of the chemistry of living organisms and molecular basis for changes taking place in plants, animals, and microbial cells. It develops the foundation of understanding all biological processes and communication within the between cells, as well as the chemical basis of inheritance and diseases in animals and plants. Chemical analysis of all living organisms indicates presence of the most common elements like carbon, hydrogen, nitrogen, oxygen, sulfur, calcium, phosphorus, magnesium, and others with their respective content per unit mass of living tissue. Chemically, all living organisms have basic three types of macromolecules, which are polymers of simple subunits called monomers. The polysaccharides, carbohydrates, polypeptides, proteins, and polynucleotides, nucleic acids, are the polymers of monosaccharides, amino acids, and nucleotides, respectively. Figure 6.1. Lipids are water insoluble and small molecular weight compounds as compared to macromolecules. Biomolecules is the cell of carbohydrates. The word carbohydrates means hydrates of carbon. They are also called saccharides. They are biomolecules made of from just three elements: carbon, hydrogen, and oxygen, with the general formula C6H2O three times. They contain hydrogen and oxygen in the same ratio as in the water 2:1. Carbohydrates and can be broken down, oxidized to release energy based on number of sugar units. Carbohydrates are classified into three types, namely monosaccharides, disaccharides, and polysaccharides. Monosaccharides, these are the simplest sugars, having a crystalline structure, sweet taste, and soluble in water. They cannot be further hydrolyzed into smaller molecules. They are the building blocks of monomers of complex carbohydrates. They have the general molecular formula CH2O 6 times, where N can be 3, 4, 5, 6, 7. They can be classified as trios, tetros, pentos, etc. according to the number of carbon atoms in a molecule, as mentioned in the table 6.2. Monosaccharides containing the aldehydes group are classified as aldoses. Example now glucose, xylose, and those with alpha ketones. Ketone group are classified as ketoses. Example ribuloses, fructose. Glucose, it is the most important fuel in the living cells. Its concentration in the human blood is about 90 mg per 100 ml of blood. The small size of solubility in water of glucose molecules allows them to pass through the cell membrane into the cell. Energy is released when the molecules are metabolized by cellular respiration. Galactose, it looks very similar to glucose molecules. They can also exhibit in alpha and beta forms. Galactose react with glucose to form the disaccharide lactose. However, glucose and galactose cannot be easily converted into one another. Galactose cannot play the same role in respect to as glucose. Fructose. Fructose, it is the fruit's sugar, and chemically it is ketohexose, but it has a 5 atom ring rather than the alpha 6 atom ring. Fructose reacts with glucose to form the sucrose disaccharide. Disaccharides. Monosaccharides are rare in nature. Most sugars, most sugars found in nature are disaccharides. Disaccharides is formed when two monosaccharides react by condensation. A reaction releasing of water molecule. This this possesses This process requires energy. The glycosidic bond forms and hold the two monosaccharides units together. Sucrose, lactose, and maltose are examples of disaccharides. So, sucrose is a non-reducing sugar since it lacks free aldehydes or ketone group. Lactose and maltose are reducing sugars. Lactose also exists in a beta form, which is made from beta galactose and beta glucose. Disaccharides are soluble in water, but they are too big to pass through the cell membrane by diffusion. They are broken down in the small intestine during digestion. Formed monosaccharides then pass into the blood and through the cell membrane into the cells. Figure 6.4 maltose. Monosaccharides Monosaccharides are used very quickly by cells, but if a cell is not in need of all the energy releasing immediately, then it may get sold. Monosaccharides are converted into disaccharides in the cell by condensation reactions, which result in the formation of polysaccharides as macromolecules. These are too big to escape from the cell. Polysaccharides. Monosaccharides can undergo a series of condensation reactions, adding one unit after the other to the chain till a very large molecule polysaccharide is formed, and this is called polymerization. Polysaccharides are broken down by hydrolysis of monosaccharides. The properties of a polysaccharide molecule depend on its length, branching, folding, and coiling. Starch is a stored food in the plants. It exists in two forms, amylose and amylopectin. Both are made from alpha glucose. Amylose is an unbranched polymer of alpha glucose, and the molecules coil into a helical structure. It forms alpha colloidal alpha colloidal suspension with in hot water. Amylopectin is a branched polymer of alpha glucose. It is completely insoluble in water. Figure 6.5 starch glycogen. It is amylopectin with very short distances between the branching side chains. Glycogen is stored in animal body, particularly in liver and muscles, from where it is hydrolyzed as per need to produce glucose. Cellulose. It is a polymer made up from beta glucose molecules, and the polymer molecules are straight. Cellulose serves to form the cell walls in plant cells. These are much together, much tougher than cell membrane. And this toughness is due to chain and hydrogen bonding between neighboring chains. Biological significance of carbohydrates. That supplies energy for metabolism. Glucose is the main substrate for ATP synthesis. Lactose, a disaccharide, is present in milk, provides energy to lactating babies. Babies. Polysaccharides serve as structural component of cell membrane, cell wall, and reserved food as starch and glycogen. Lipids. These are group of substances with greasy consistency with long hydrocarbon chains containing carbon, hydrogen, and oxygen. In lipids, hydrogen to oxygen ratio is greater than 2 to 1. In carbohydrates, it is always 2 to 1. A lipid is a broader term used for fatty acids and and their derivatives. They are soluble in organic solvents, non-polar solvents. Let's understand what fatty acids are. Fatty acids are organic acids which are composed of hydrocarbon chain coding ending in carboxyl group. They can be saturated fatty acids with no double bonds between the carbon atom and the of the hydrocarbon chains. Palmitic and stearic acids found in all animal and plant fats are examples of saturated fatty acids. Unsaturated fatty acids are with one or more double bonds between the carbon atom of the hydrocarbon chains. So they are oleic acid found in nearly all fats and linoleic acid found in many acid oils are examples of unsaturated fatty acids. These fatty acids are basic molecules which form different acids of lipids. Lipids may be classified as simple compound and derived lipids. Simple lipids. These are the esters of fatty acids with various alcohols, fats, and waxes are simple lipids. And fats are esters of fatty acids with glycerol. Triglycerides are there are three molecules of fatty acids and one molecules of glycerol. Generally, unsaturated fats are lipids at room temperature and are called oils. Unsaturated fatty acids are hydrogenated to produce fats. Example: vanaspati ghee. Fats are the nutritional source with high calorific value. Fats act as reserved food materials in plants. It is stored in seeds to nourish embryo during germination. In animals, fat is stored in the adipose sites of the adipose tissue. The fats deposited in subcutaneous tissue act as an insulator and minimize loss of body heat. Fats deposited around the internal organs act as cushions to absorb mechanical shocks. Wax is another example of simple lipid. They are esters of long chain fatty acids with the long chain alcohols. They are found in blood, gonads, and sebaceous glands of the skin. Waxes are not as readily hydrolyzed as fats and waxes and are solid or ordinary temperature. Example: wax in bitu, ox in bitu. Waxes form water insoluble coating on hair and skin in animals. Waxes form out an outer coating on stems, leaves, and fruits. Component lipids. These are the easier ester of fatty acids containing other groups of phosphate, phospholipids, sugar, glycolipids, etc. They contain the molecule of glycerol, two molecules of fatty acids, and a phosphate group of simple sugar. Some phospholipids, such as lecithin, also have a nitrogenous compound attached to the phosphate group. Phospholipids have both hydrophilic polar groups, phosphate and nitrogenous group, and hydrophobic non-polar groups, hydrocarbon chains of fatty acids. The phospholipids contribute in the formation of cell membrane. Glycolipids contains glycerol fatty acids, simple sugars such as galactose. And they are also called cerebrosides. A large amount of them have been found in the brain. White color matter and myelin sheath. Figure 6.9 lipid bilayer and in aqueous medium sterols. They are derived lipids. They are composed of fused hydrocarbon rings, steroid molecules, steroid nucleus, and a long hydrocarbon side chain. One of the most common steroid in cholesterol. It is widely distributed in all cells of the animal body, but particularly in in the in the nervous tissue. Cholesterol exists of either 5 either 5 or as cholesterol, ester, adrenocorticoids, sex hormones, progesterone, testosterone, and vitamin D are synthesized from cholesterol. Cholesterol is not found in plants. In plants, cholesterol exists chiefly as phytosteroids. Yam plants for product produces steroid compound called diosgenin. It is used in the manufacture of anti-fertility pills, that is birth control pills. Proteins, the term protein, proteinous, meaning first of the primary importance, was suggested by Berzelius 1830. Mulder adopted the term protein to refer to the complex organic nitrogenous substances found in the cell of the all animals and plants. Characteristics: proteins are large molecules containing amino acid units ranging from 100 to 3000. Proteins have high molecular weights. In proteins, amino acids are linked together by peptide bonds which join the carboxyl group of another residue. A protein molecules consists of one or more polypeptide chains. Now, proteins can contain any or all of the 20 naturally occurring amino acid types. The linear sequence of amino acids in polypeptide chain of a protein forms its primary structure. Functional proteins have three-dimensional conformation. Some proteins, such as keratin, of hair, consist of polypeptide chain arranged like a spiral helix. Such spirals are in some cases right-handed, called alpha helix. In others, left-handed, called a beta helix. The spiral configuration is held together by hydrogen bonds. The sequence of amino acids in the polypeptide chain also determines the location of its bend or fold and the position of formation of hydrogen bonds between different portions of the chain or between different chains. Due to formation of hydrogen bonds, peptide chains assume a secondary structure. In some proteins, two or more peptide chains are linked together by intermolecular hydrogen bonds. Such structures are called plated sheet. Plated sheet structure is found in protein of silk fibers. In large proteins such as myoglobin and enzymes, peptide chains are much looped, twisted, and folded back on themselves due to formation of disulfide bonds. Such loops and bends give the protein of tertiary structure, whereas in hemoglobin, protein subunits are held together to form a quaternary structure. Proteins are extremely reactive and highly specific in behavior. Proteins are amphoteric in nature, that is, they act as both acids and bases. The behavior of proteins is strongly influenced by pH. Like amino acids, proteins are the dipolar ions at the isoelectric point, that is, the sum of the positive charges is equal to the sum of the negative charges, and the net charge is zero. The ionic groups of protein are contributed by side chains of the polyfunctional amino acids. A protein consists of more basic amino acids such as lysine and arginine exist as a cation at the physiological pH of 7.4. Such proteins are called basic proteins. Histones of nucleoproteins are basic proteins. A protein is rich in acidic amino acids exists as an anion at the physiological pH. Such proteins are called acidic proteins. Most of the blood proteins are acidic proteins. Classification of proteins on the basis of structure. Proteins are classified into three categories. Simple proteins. Simple proteins are on hydrolysis yields only amino acids that are soluble in one or more solvents. Simple proteins may be soluble in water. Histones of nucleoproteins are soluble in water. Globular molecules of stones are not coagulated by heat. Albumins are also soluble in water, but they get coagulated on heating. Albumins are widely distributed. Example: egg albumin, serum albumin, and legumin of pulses and albumins. Proteins. Conjugated proteins consist of a simple protein united with some non-protein substances. The non-protein group is called a prosthetic group. Example: hemoglobin. Globin is a protein, and the iron containing pigment hem is the prosthetic group. Similarly, nucleoproteins have nucleic acids coupled with the histone protein. On this basis, proteins are classified as glycoproteins and mucoproteins. Mucoproteins are carbohydrate proteins. Complexes are example mucin of saliva and heparin of blood. Now, lipoproteins are lipid protein complexes. Example: conjugate protein found in brain, plasma membrane, milk, etc. Derived protein. These proteins are not found in nature of such. Now, these proteins are derived from native protein molecules and hydrolysis metaproteins, peptones, and derived proteins. Nucleic acids. Fuel Fuel 1924 showed that chromosomes contain DNA. They established that nucleic acids contain two polypyrimidine, cytosine, and thymine. The two purine adenine and guanine bases. Wilkins and co-workers showed that the purine and pyrimidine bases are placed regularly along the DNA molecules at a distance of 3.4 angstrom. DNA is composed of sugar molecules. Now it is the pentose sugar of deoxyribose type. Phosphoric acid, also called phosphates, when in chemical combination nitrogen are containing bases. These are nitrogen containing organic ring compounds. Principally bases are of two types: pyrimidine bases and purine bases. Pyrimidine bases are a single ring monocyclic. Nitrogenous bases cytosine, thymine, and uracil are pyrimidines. Purine are double ring dicyclic. Nitrogenous bases adenine and guanine are purines. Irwin Chargaff 1950 estimated the relative amounts of the four nitrogenous bases by adenine, thymine, cytosine, and guanine and DNA. They observed that the pyrimidine and purine always occurs in equal amount in DNA. They also found that the base ratio, that is A plus T upon G plus C, they may vary in the DNA of different or groups of animals and plants, but A plus T upon G plus C ratio remains constant for a particular species. Structure of DNA. DNA is a very long chain made up of alternate sugars and phosphate groups. The sugar is always deoxyribose, and it always joined to the phosphate in the same way, so that the long chain is perfectly regular, repeating the same phosphate sugar sequence over and over again. Each sugar is of the sugar phosphate chain has a base attached to it, and the base is not always the same. This unit, which consists of sugar, phosphate, and a base, is called a nucleotide. The nitrogenous base and the sugar of a molecule nucleotide form on a molecule nucleotide. Thus, nucleoside does not contain phosphate group. Four types of nucleosides are found in DNA molecules. In a nucleoside, nitrogenous base is attached to the first carbon atom C1 of the sugar, and when the phosphate group gets attached with that of the carbon C5, atom of the sugar molecule. A nucleotide molecule is formed. Figure 6.10 nitrogen bases in nucleic acids. A single strand of DNA consists of several thousands of nucleotides, one above the other. The phosphate group of the lower nucleotide attached with the fifth carbon atom of deoxyribose. The sugar forms phosphodiester bond with that of the third carbon atom of the deoxyribose sugar of the nucleotide placed just above it. Figure 6.11 Structure of DNA. Single long chain of polynucleotides of DNA consists of one end of with sugar molecules not connected with another nucleotide having C3 carbon and not connected with the phosphate group. Similarly, the other hand having C5 of the sugar C5 of the sugar is not connected with any more phosphate group. These two ends of the polynucleotide chain are called as 3' to 5' 3' and 5' ends respectively. The single polynucleotide strand of DNA is not straight but helical in shape. The DNA molecule consists of such two helical polynucleotide chains which are complementary to each other. The two complementary polynucleotide chains, the DNA are held together by the weak hydrogen bond. Adenine always pairs with thymine and guanine with the cytosine. A pyrimidine with a purine. Adenine thymine pair consists of two hydrogen bonds and guanine cytosine pairs. Sequence of bases of polynucleotide chain is known. That of the other can be determined in a model. Ribonucleic acid RNA. And another nucleic acid found in the living organisms is ribonucleic acid. In most of the living, it is not found to be hereditary material, but in certain organisms like tobacco or mosaic virus, it is hereditary material. Like DNA, ribose nucleic acid also consists of polynucleotide chain with the difference that it consists of single strand. In some cases, example reovirus and wound tumor virus, RNA is double stranded. The nucleotides of RNA have ribose sugar instead of the deoxyribose sugar, as in the case of DNA. Figure 6.12 single strand of DNA RNA. In case of RNA, uracil substitutes thymine of DNA. Purine pyrimidine equality is not found in the RNA molecules because of its single stranded structure. RNA strand is usually found folded upon itself and in certain regions and entirely. These foldings help in stability of the RNA molecules. Most of the RNA polynucleotides chains start either with the adenine and guanine. Three types of cellular RNAs have been distinguished: messenger RNA, mRNA, template RNA, ribosomal RNA, rRNA, transfer RNA, tRNA, or soluble RNA. mRNA carries genetic information for arranging amino acids in the definite sequence. It is it is a linear polynucleotide. It accounts 3% of cellular RNA. Its molecular weight is several millions. mRNA molecule carrying information to form a complete polynuclear polypeptide chain is called cystine. Size of mRNA is related to the size of message it contains. Synthesis of mRNA begins at 5' end of DNA strand and terminates at 3' end. rRNA form 50 to 60% or part of the ribosomes. It accounts 80 to 90% of the cellular RNA. It is synthesized in nucleus. Kirtland 1960 discovered it gets coiled here, and they are due to interest chain complementary base pairing. Figure 6.14, the RNA. tRNA molecules are much smaller, consists of 70 to 80 nucleotides. It is also single or stranded, but no to number of complementary base sequences. After pairing, it is sugar, clover leaf, holly. 1965, each tRNA can pick up particularly amino acids. Following four parts can be recognized, and tRNA1, DHU dehydroxyuracil loop, amino acid recognition site, amino acid binding site, anticodon loop, codon recognition site, ribosome recognition site. Within the anticodon loop of tRNA, three unpaired nucleotides are present, called as anticodon, which pair of within codon present on mRNA. The specific amino acids is attached at the 3' end is accepted them of clover leaf of tRNA. Enzymes. Properties of enzymes. Nature of enzymes. Properties of enzymes. Proteinaceous nature, three-dimensional conformation, catalytic properties, specific action, temperature, nomenclature of enzymes, classification of enzymes, concentration of substrate, enzyme concentration, temperature, effect of pH, other substances, catabolic pathways, anabolic pathways, metabolic pool, secondary metabolites, terpenes, phenols, नायट्रोजन कंटेनिंग कंपाऊंड्स. तर इथं आपला संपतो बायोमॉलिक्युल्स हा चॅप्टर, जो आपण थोडक्यात आटोपला इथे. हा चॅप्टर खूप मोठा चॅप्टर आहे, तर आपण हा थोडक्यात इथं कव्हरअप केलेला आहे. त्यानंतर आपण एक्झरसाईज दुसऱ्या व्हिडिओमध्ये पाहणार आहोत. तरी प्लीज या चॅनलवर तुम्ही जर नवीन असाल तर प्लीज या चॅनलला सबस्क्राईब करून ठेवा, जेणेकरून तुम्हाला आमचे नवीन नवीन व्हिडिओज पाहायला मिळतील. चला तर मग, थँक्यू सो मच फॉर वॉचिंग. प्लीज लाईक, शेअर अँड सबस्क्राईब.